Hello viewers, this is Arnabha Chatterjee back with a new video on human reproduction. It's a very important topic for this physiological discussion on biology and for clearing the essential entrance examinations, including the CSI and NET examination. So I'll be discussing two chapters, and please be very attentive and please follow me. So I'll be reading out, and I am actually following my class 12 biology book, which is a very good book, because in all the honors books and in the books which I have studied for post graduation like MSc biotechnology, I didn't find any good like complete concrete material as human reproduction given in any of the books in my honors and postgraduate courses than in this class 12 biology book, which is actually Nutan's biology book by V. Singh and D.K. Jain, which is of Nagin's Prakashan. It is actually ICSC and ISC biology publication books. So since I was a student from ISC background, like ISC board, so I had this biology book, and it's a very beautiful book. It explains everything in lucid and clear language. So I'll be reading out from this book. So, no organism survives forever, and therefore, if a species is to continue to exist, its member must reproduce their own kind. Reproduction is a process that results in the formation of new individuals of the same kind, though usually with slightly genetic, structural, and physiological variations. Because we know that the DNA from father and from mother intermingles, and a new variety of organism is produced. This is actually the basis of sexual reproduction. The chief objective of reproduction is therefore perpetuation of the species and consequently increase in the number of individuals. So we have some patterns of animal reproduction. Among animals, there are two types of reproduction, asexual and sexual. Asexual reproduction does not involve formation or union of sex cells or gametes, whereas sexual reproduction involves the formation and fusion of sex cells. The latter leads to the creation of new combinations of genes by pooling together in one line of descent the genes derived from two different parents. This results in a reshuffling of the gene material. The recombinants so produced can then be subjected to natural selection. Thus, sexual reproduction involves the continuation of nuclear material and frequently cytoplasm from two organisms of the same species. So, we are having some differences between sexual and asexual reproduction. In asexual reproduction, a single organism is capable of producing its own kind, that is, participation of two organisms is not required, and the individuals produced are genetically identical. It ensures rapid multiplication and is especially useful where an organism needs to colonize a localized area in a short time. It is mostly a characteristic feature of lower organisms such as protozoans, sponges, coelenterate, certain flatworms, etc. Sexual reproduction. Most animals reproduce sexually. In sexual reproduction, development of new individual takes place by the fusion of two sex cells, the male and female gametes. The zygote formed by the fusion of male sperm and female ovum gametes gives rise to a multicellular embryo by repeated mitotic divisions. The embryo gradually develops into a full-grown individual. Sexual reproduction is energetically very expensive because large number of gametes must be generated, of which only a few are used to form zygotes, which grow to reproductive maturity. The advantage of sexual reproduction is the generation of variation upon which natural selection can act. In higher animals, like sperms and ovas, are produced in specialized sex organs such as gonads. The male gonad, called testis, produces sperm, male gametes, and the female gonad, known as ovary, produces ova, that is the female gametes. In most animals, sexes are separate That is, an individual possesses either testes or ovaries. These animals are thus unisexual. But in some animals like earthworm and tapeworm, the same individual carries both testes and ovaries. Such animals are called bisexual or hermaphrodites. In bisexual organisms, invariably cross-fertilization takes place. Okay, so let us have a look at the mammalian reproductive system. Mammals have separate sexes, male and female. The primary sex organs in males are testes and in female ovaries. Besides, there are reproductive ducts and some accessory structures. So male reproductive system is, in humans, the male reproductive system consists of the following things: a pair of sex glands known as testes, a duct system comprising vasca, efferentia, epididymis, ductus deferens and ejaculatory ducts, accessory glands such as seminal vesicles, prostate and bulbolateral glands, copulatory organ, the penis containing urethra. The male sex cells, spermatozoa, are produced by testes, conducted by various glands and ejected through urethra. A fluid is produced by the secretions of the glands which nourish spermatozoa during the passage. The secretion of glands with spermatozoa is a whitish viscous fluid known as seminal fluid or semen. The process of expulsion of semen from the urethra is called ejaculation. So sperms are produced in a pair of glands called testes. In an adult male, the testis is an oval structure about 4 cm long and weighing about 25 gram. So location: the testes of a male remain suspended in a pouch called scrotum outside the abdominal cavity. They actually develop in the abdominal cavity near the kidneys but migrate to the scrotum shortly before or at the time of birth. Sperm development occurs optimally at temperatures a few degrees below the normal body temperature of 37 degrees centigrade. So sperm development actually occurs between a optimum temperature of 32 to 33 degrees centigrade. As scrotum has little or no fatty insulation, it keeps the testis as a temperature cooler than the body temperature. Moreover, a suitable temperature is maintained inside the testis by involuntary movements of the muscles that connect the scrotum to the body wall. So the sphincter's muscles are actually responsible for this maintenance of optimum temperature. Contraction or relaxation of muscles moves the scrotum nearer to or further away from the heat of the body when environmental temperatures are low or high, respectively. However, in some seasonally breathing animals, testes descend into the scrotum in the breathing season but ascend back into the abdomen in the non-breathing state, as in case of dogs and cats. Okay. Structure: the outermost layer of the testis is a dense fibrous structure called tunica albuginea. Internally, each testis is divided into several lobules. Each lobule contains a number of tiny, highly convoluted tubules known as seminiferous tubules. Each tubule is 70 to 80 centimeter in length when stretched out. The basement membrane of the seminiferous tubule is lined by highly specialized cells called spermatogonia. Spermatozoa occupy the central part of the lumen of the seminiferous tubule. Another type of cell, known as cells of Sertoli, are also attached to the basement membrane. These cells provide mechanical support and protection to the developing germ cells and also participate in the nutrition and maturation. In between seminiferous tubules, masses of cells called interstitial cells or Leydig cells are present. These cells secrete the male hormone testosterone, which is responsible for the development and maintenance of male sex characteristics. The interstitial connective tissue also contains fibroblasts, blood vessels, nerves and lymphatics. Duct system. The duct system consists of tubules which conduct sperms from the testis to the outside. The seminiferous tubules open into a structure known as rete testis. Several vasa efferentia arise from the rete testis, which open into the epididymis and conduct spermatozoa out from the testis. The epididymis is long about 6 meters when fully uncoiled but highly convoluted tube resting upon and on the side of the testis. It has a head, a body and a tail. The head of the epididymis is made up of highly convoluted continuations of the efferent ductiles or the ductules. At the lower end of the head, these tubules join to form a single tube called the duct of epididymis. This duct is highly coiled on itself and forms the body and tail of the epididymis. At the lower end of the tail, the duct becomes continuous with the ductus deferens. The epididymis temporarily stores spermatozoa, which are metabolically inert, non-motile and mature. Storage in the epididymis makes them motile and mature. A partially coiled thick muscular tube from each epididymis ascends into the abdomen. This tube, known as ductus deferens, conducts spermatozoa from the epididymis to the base of the urinary bladder. The ductus deferens joins the duct of the seminal vesicle at the base of the urinary bladder to form the ejaculatory duct, which opens into the prostatic part of the urethra. The terminal dilated part of the ductus deferens is called ampulla. Okay. The urethra is about 18 to 20 centimeter long. From the urinary bladder, it passes through the prostate glands and leads to the penis. So we are having these things. So we will be dealing with the male and the female reproductive organs, and we will be dealing with the menstrual system in later videos. Do keep posting your valuable comments, and please support me on my YouTube, LinkedIn, and other channel and other platforms. Thank you so much.